Hi everyone, my name is Sarah Hammerland and I'm a PhD student in the Department of Ecology, Evolution, and Behavior at the University of Minnesota. And in this video, I'm going to tell you a little bit about the microbial evolution research area.
My research is in the field of microbial experimental evolution. I work with bacteria because they evolve fast. That's because they grow rapidly with generation times as short as 20 minutes, and they have huge population sizes. There are more E. coli cells in this test tube than there are humans on Earth. Bacteria are also pretty simple to work with. I can easily grow my bacteria in different environments, dilute and plate my cultures to figure out their population densities, and I can directly measure the relative fitness of different strains by pitting them head-to-head in competition assays. I can also stick my bacteria into the -80-degree freezer, leave them there for as long as I want, and then come back later to revive them. This is like a living fossil record, and it means I can let my bacteria evolve for many generations, revive their ancestors, and then directly compare them. In this research area, students will be using experimental evolution with bacteria to investigate a major public health problem. You may have heard about so-called superbugs, which scientists and public health experts are really concerned about. Maybe you know someone who's had a staph infection that was very hard to treat. Because bacteria evolve quickly, and because antibiotics pose a very strong selection pressure, it's very common for bacteria to gain mutations that make antibiotics useless. Students who work in this research area will subject populations of the bacteria E. coli to the antibiotic rifampicin. You'll explore fundamental questions about evolution and natural selection and gather data about how quickly E. coli become resistant to rifampicin. You'll learn how to measure the level of rifampicin resistance, which will look a bit like this culture plate containing E. coli and different concentrations of rifampicin. You'll also sequence a gene where resistance mutations commonly occur. You'll have the freedom to formulate your own hypotheses and apply what you know about evolutionary biology to this powerful experimental system. You'll also learn how to use sterile technique to keep pesky contaminants out of your growth media in petri dishes. Though, honestly, contamination is sometimes just part of the life of a microbiologist.
I think the microbial evolution research area is a great option for anyone interested in evolutionary biology, ecology, public health, medicine, or if you just want to play around with some bacteria.